Hello everyone, welcome to our channel Bioskills Circle, and in this video we will talk about gram staining, right? So gram staining is a method of staining used to distinguish and classify bacterial species into two large groups, and you can see here that bacteria can be categorized into two large groups, that is gram positive bacteria and gram negative bacteria. The name comes from Danish bacteriologist Hans Christian Gram, who developed this technique. So, gram staining is a differential technique used to distinguish two large groups of bacteria based on their cell wall constituents. Let me tell you here that the main difference between gram positive and gram negative bacteria is their cell wall composition. The gram positive bacteria, you can see here, they have thick layer of peptidoglycan, whereas gram negative bacteria, they have thin layer of peptidoglycan, right? And gram positive bacteria have teichoic acid present in their cell wall, whereas gram negative bacteria lack teichoic acid. Moreover, gram negative bacteria, they have additional lipopolysaccharide layer. So the structure of organism cell wall determines whether the organism is gram positive or gram negative. Let us talk about reagents now. Okay, so the first reagent in gram staining is crystal violet. Crystal violet is also called primary stain. Second reagent is iodine. Iodine acts as a mordant. What is mordant? Mordant is an agent that fixes crystal violet to bacterial cell wall. Third is alcohol. Alcohol acts as a decolorizer, right? And it is made up of acetone and alcohol. This decolorizer, this is either made of alcohol or acetone. Then fourth reagent is safranin, this one, and this acts as a counter stain. So how does gram staining work? You need to remember here that it involves three main processes. First is staining with crystal violet, then decolorization, that is done with alcohol, and third step is counter staining, that is usually done with safranin. Let's talk about the procedure now. First step is fixation, right? In this step, we fix material on slide with methanol or heat. If slide is heat fixed, allow it to cool for some time before applying the stain. In the second step, cells are stained with crystal violet dye. Now this crystal violet dye dissociates into CV positive and chloride ions in aqueous solution, and these ions penetrate the cell wall of gram positive and gram negative bacteria. The CV positive ion interacts with negatively charged components of bacterial cells and stains cell purple. So you can see here that both gram positive bacteria and gram negative bacteria stain purple, right? Then the next step is iodine treatment. As I told you earlier that iodine acts as a mordant, right? So in this step, iodine solution is added to form larger complex between crystal violet and iodine. And the fourth step is decolorization. So a decolorizer such as ethyl alcohol and acetone is added to the sample. Since gram-negative bacteria have thin peptidoglycan layer and have additional lipopolysaccharide layer which gets dissolved due to addition of alcohol, so bacteria gets decolorized as the complex is washed away. So you can see that gram-negative bacteria after this step, right, they appear colorless, right? So we can say here that gram-negative bacteria, because they have thin peptidoglycan layer and additional lipopolysaccharide layer, so because of the action of alcohol, these layers they get dissolved, right, and bacteria gets decolorized as the complex is washed away. In contrast, gram positive bacteria, you will see after this decolorization step, they remain purple here, right? So a gram positive bacterium or cell becomes dehydrated from the ethanol treatment. This closes the pores in the cell wall and prevents the stain from exiting the cell, right? The large crystal violet iodine complex gets trapped within the cell wall of gram positive bacteria here. So you can see here after decolorization, the gram positive cell remains purple and gram negative cell loses its purple color. So the bacteria appear colorless here, right? And the final step is counter stain with safranin. So a counter stain, which is usually positively charged safranin, is added to the sample. Since safranin is lighter than crystal violet, it does not disrupt purple coloration in gram positive cell, right? So here they remain purple. However, the decolorized gram negative cell, they are stained red, right? So to sum up here, gram positive bacteria stain purple due to the presence of a thick layer of peptidoglycan in their cell wall which retains crystal violet, whereas gram negative bacteria, they stain red, which is attributed to thinner peptidoglycan wall which doesn't retain crystal violet during decolorizing process.